In this video, we are going to explore the metabolomics workflow. In other words, if you were aiming to determine the differences in the metabolome between a treatment group and a control group, meaning a group of people with a disease versus a group without, a group of organisms that are under a particular stress versus ones that don't have that stress, or comparing metabolites across the full age range, or a variety of other applications, how would you go about those metabolomics analyses? So we're going to walk through the full process of this, beginning with the collection of samples and extraction of those samples through the analysis of compounds within those samples by LCMS, GCMS, NMR, and other techniques. And then we will give an overview of the ways that those really complex data sets are analyzed and interpreted. So let's go ahead and take a look at this metabolomics workflow from the beginning. By the end of this, you should be able to provide an overview of a general metabolomics workflow and apply that toward answering questions related to conducting metabolomics experiments. What I'm going to do is break down the metabolomics workflow one step at a time. So the first step of the metabolomics workflow is actually collecting the sample and extracting that sample. So I'll go ahead and put in here my first step of the process is just the collection of the biological sample. And this could be a collection of a urine sample, it could be a collection of a blood sample, it could be the collection of bacteria from a bacterial culture that is growing on a plate or in a broth of liquid media. Whatever your biological system is, that's going to determine what your sample collection is. And generally, the sample collection is not just from a single treatment organism and a single control organism, but it's generally relatively large numbers of replicate treatments and replicate controls, meaning oftentimes 10 plus replicate treatments and 10 or more replicate controls. The reason why such a large number of replicate treatments and controls is often required is because of the fact that there is a large amount of inherent variability when working with biological systems. And so that inherent biological variability needs to be able to be differentiated from actual tractable changes that result from treatments so that we are able to make clear conclusions about what changes are actually statistically significant versus what observations we might be making just based on random chance and random variability from one biological sample to another. So the sample collection would be the collection of things like urine, blood, saliva, plant tissue, bacteria cells from a liquid culture or growing on a petri dish, whatever your choice might be. So a variety of different possible options there. Whatever sample is collected, the next step in the process is generally that sample can't be directly analyzed by LCMS, NMR, or some other analytical technique. Instead, the small molecule metabolites have to be extracted from that sample because the sample contains a huge variety of different biomolecules, including both the small molecules that we look at in metabolomics and macromolecules such as nucleic acids, proteins, etc. And so generally, what happens after sample collection is the extraction of the small molecules. And remember that in the last video, we defined small molecules as generally ones that have a molecular weight of about 1.5 kilodaltons or less. So our extraction here, the purpose of this is to extract, or in other words, separate the small molecules. And generally we're focusing on small organic molecules from our biological macromolecules, because analysis of those biomacromolecules such as proteins or DNA would fall under the headings of genomics or proteomics rather than metabolomics. So we're looking at the small molecules. And so extraction takes place here of both the treatment samples and the control samples. So I'm going to put treatment versus control samples that you have collected would be subjected to the extraction process. And there are a variety of ways to extract small molecules from samples. And the exact extraction method is largely dependent upon exactly what type of organism are you working with. Are you working with a plant versus are you working with human urine or bacteria in culture? Some common ways of going about conducting extractions of the metabolome involve, if we were working with, say, a plant, which I will draw my best rendition of a plant, which don't get too excited because my best rendition of a plant is pretty awful. But if we had a plant, what we could do to extract the small molecules from that plant was we could put the plant into a container such as an Erlenmeyer flask, and we could soak that plant with an organic solvent. So I'll say that this organic solvent in here that I've drawn in as a squiggly line is methanol, a common organic solvent. And what methanol, the organic solvent, will do is it will solubilize the small organic molecules, extracting them from the plant cells, extracting them from that plant tissue that is there. And so ultimately, the compounds that are present within the plant will be introduced into that methanol solvent. And so what the methanol is doing here is it is serving to help lyse open the plant cells and solubilize the organic metabolites, the small molecules in that organic solvent. And then what would happen from there in this extraction process in methanol is that generally the sample would be filtered, and that would remove the insoluble material, such as there will always be some plant debris left. And that would also help remove things like proteins and other macromolecules. And then that would result in a chemical extract, as we would call it, in the methanol solvent, which I'll abbreviate methanol as MEOH. And then what could be done is that this chemical extract could be placed into a low pressure environment to enable the evaporation of that volatile organic solvent, methanol. And so in other words, what would happen is that the methanol would be generally removed in vacuo, meaning under vacuum, so that you would be left with a chemical extract from that plant, which would be a dried residue of the assortment of small molecules that were present in that plant and extracted using the methanol solvent. On the other hand, as an alternative to that, if instead one were working with a sample that is liquid, such as urine or perhaps saliva or bacteria that's growing in a liquid culture, commonly, a technique that's used for the extraction of compounds for those metabolomics analyses is what's referred to as liquid-liquid extraction. So this is my best attempt at a separatory funnel that I've drawn there in black. And liquid-liquid extraction, also referred to as solvent partition, is a technique that you learned back in Organic 1 or Organic 2 lab. And what happens in that technique is that your initial sample would generally be present in aqueous material, such as urine is an aqueous substance, saliva, bacteria growing in liquid culture. So you would take that aqueous material that contained your compounds of interest, and you would mix that in this separatory funnel with an organic solvent that is immiscible with the aqueous. So this would be an organic solvent, and it can't be just any organic solvent. It has to be immiscible with the aqueous layer, meaning that it will not solubilize in the aqueous material. Instead, it will separate out as two separate layers in the separatory funnel. So generally what happens here is that you would start by pouring the sample of interest, such as the urine sample, into the separatory funnel, add the organic solvent. You would shake this to extract the organic molecules from the aqueous layer into the organic layer, and then you would drain out and discard the aqueous material, keep the organic material, and that organic material then could be dried in vacuo to remove the solvent, leaving a residue of the mixture of compounds that was present in that urine sample or whatever your biological sample was here. So after the liquid-liquid extraction, generally what happens, analogously to the extraction of cell systems and methanol that we saw up top here, was that the organic solvent is removed in vacuo, leaving us with a dried residue of the organic compounds. And the organic solvent that is selected here can be a variety of different solvents. The main criteria is that it can't be able to be forming a single solution with water. It has to be able to be separated from water. And so common organic solvents here are things like ethyl acetate, an ester, hexane, or other alkanes, and a variety of other organic solvents as well, such as chloroform, dichloromethane, and others. In each of these extraction techniques, it is important to point out that there are limitations. And a limitation to any extraction technique is that there will be some inefficiency, some compounds that are not extracted particularly well into the organic solvent. And ultimately, if the compound is not extracted well into the organic solvent, it is not going to be available later on downstream for detection by LC-MS, NMR, or any of our other techniques. And so as a result, some compounds can be overlooked in metabolomics because of limitations in this workflow. So at the stage of the extraction, a limitation here is that not all organic molecules will be extracted into the organic solvent. For example, if we had a relatively polar organic molecule, when we did the liquid-to-liquid extraction, that could very well end up in the aqueous layer there, and that would then be missed downstream. And so as a result, when we use the term metabolomics and we define it as looking at all of the metabolites that are present within a cell, that's a bit of a misnomer because some of the compounds are going to be missed as a result of limitations at different stages of the workflow here, with one of those stages where the compounds can be overlooked or missed is at the extraction stage because the compound simply is not well extracted into the organic solvent. So in practical terms, what this means is if you are working through a metabolomics workflow, if you hypothesize that the compounds that you are most interested in are ones that are relatively polar, then you need to focus on extraction methods that will bias toward polar compounds. On the other hand, if you are interested primarily in compounds that are less polar, you hypothesize those will be the most important in what you're trying to observe in your metabolomics analysis. Then focus on the less polar solvents, the more non-polar solvents. And if you're not sure at all, another thing that is sometimes done is that people will run multiple types of chemical extractions and conduct metabolomics analyses on all of those chemical extracts. That can get pretty laborious though. So let's go on to the next step of the metabolomics workflow here. So the next step of the metabolomics workflow, after the compound mixture has been extracted from the biological system, then what next typically happens is that complex mixture containing potentially thousands of different organic molecules is separated to simplify the mixture for analysis by mass spectrometry or NMR. So our next stage of this we'll refer to as our mixture separation. And this is a step that is generally done in metabolomics, although I will say it is not always done. So we could consider this to some extent to be an optional step because there are some techniques available for NMR in particular that will enable the analysis of very complex NMR spectra that feature many different compounds. But as a general rule, the complex mixture is typically most commonly separated before it is evaluated by mass spectrometry or NMR in the metabolomics workflow. And so the two main techniques that are applied for the separation of complex mixtures are liquid chromatography, specifically HPLC, high performance or high pressure liquid chromatography, which is generally in these workflows just referred to as LC for liquid chromatography to shorten that up a bit. This is the same exact technique that we talked about last week when we were referring to LCMS, where we passage the compound mixture through a chromatography column that separates the compounds based on some physical property such as polarity, so that when the compounds exit the column, they're exiting the compound at different time points based on their polarity or some other physical property, and then entering the mass spectrometer or NMR for data acquisition. Another technique that is used to separate mixtures is, rather than liquid chromatography, gas chromatography, which I will write out that gas chromatography stands for GC since we haven't talked about that before. Gas chromatography is becoming less and less prevalent in the world of metabolomics because gas chromatography is more limited in its applicability. Gas chromatography separates compounds that are present in the gaseous state, and as a result requires that compounds be sufficiently volatile in order to transition to the gaseous state during the separation process, because the compounds are separating based on their different boiling points, the temperature at which they transition from liquid to gas. So gas chromatography requires that compounds are volatile, meaning they are able to transition from the liquid state into the gaseous state. And that is certainly not applicable for all organic molecules. In fact, a variety of organic molecules simply decompose when they are subjected to heat. And so gas chromatography is limited in that we have to be looking at the subset of molecules that are expected to be volatile. Plants very typically produce volatile compounds. We can recognize those qualitatively with our noses because we notice that plants often have very fragrant smells. The reason they have fragrant smells is due to the volatile compounds they're producing. So in the world of plant metabolomics, GC is a relatively commonly applied method for separating compounds from a mixture before analysis by mass spectrometry or NMR. But generally, HPLC or LC is a more common method used to separate the organic molecules. After the compounds of the mixture are separated by LC or GC, what then occurs is the acquisition of our analytical data, typically by mass spectrometry or NMR. And it's important to note here that although I'm separating out the separation of mixtures from the acquisition of the analytical data, these two steps are often completed online, meaning with a single instrument that does both of these techniques in a completely hands-off fashion. So we'll go ahead and add to our next list in the lineup here the analytical data acquisition. Putting a box around that and keeping in mind that the separation of the mixture and the analysis of the acquisition of the data, such as mass spec or NMR data, are typically conducted online, meaning with a single instrument where one would start by injecting the sample into the system. It would pass through the LC and then immediately into the mass spectrometer or NMR without you physically having to do these as two separate steps. So in the analytical data acquisition phase, generally those data are mass spectrometry, MS, or NMR, which we talked about both of these techniques last week. When we refer to NMR, most commonly it is going to be proton NMR that is applied in metabolomics analyses because proton NMR spectroscopy is more sensitive than carbon NMR spectroscopy because of the fact that the nuclear magnetic resonance active hydrogen is hydrogen 1, the most common isotope, whereas the NMR active carbon is carbon 13, and that carbon 13 isotope is definitely not the most naturally prevalent type of carbon. And so As a result, proton NMR is most commonly used for the analytical data here. Occasionally, two-dimensional NMR data are also collected here, such as HMBC data or COSY data, to enable additional information about the structures of molecules that differ between treatments and controls. Mass spectrometry can be linked to either the LC or the GC. And the mass spectrometry methods here include all of the different techniques that we talked about in our earlier videos for ionizing compounds, such as electron ionization or electron impact, electrospray ionization, and MALDI, as well as a variety of others. So when we think about the analytical data acquisition here, there are some pros and cons to NMR versus mass spectrometry that we would need to consider. And we can summarize those pros and cons as follows and design metabolomic experiments that are best suited to work around these pros and cons. So when we think of NMR as our detection method, NMR will detect any molecule that has carbons and hydrogens, and hence we could think of NMR as being really a universal detector of organic molecules. On the other hand, comparing that with mass spectrometry requires ionization, and so hence it is not universal because not all molecules will ionize equally well. For example, when we were speaking of mass spectrometry before, we talked about how compounds have to be able to pick up a positive or negative charge, and that often occurs much more capably for some functional groups than for others. And so as a result, the mass spectrometry detector is far from universal. Instead, it requires ionization, and often that ionization has to be optimized for specific classes of biomolecules. Whereas NMR, if we're conducting a proton NMR experiment, the proton NMR experiment is going to detect any protons within that particular sample. On the other hand, mass spectrometry has the key advantage that mass spectrometry has a superior limit of detection, which I'll abbreviate limit of detection as LOD. And as a result of having a superior limit of detection relative to NMR, that implies that less sample is required. And when thinking about these metabolomics experiments and metabolomics workflows, often the amount of sample is very limited. For example, if one is looking at blood samples, patients are not going to be particularly keen on providing really large blood samples. So the less, the better. And mass spectrometry can deal with generally a much smaller amount of sample because of the fact that it has a very superior limit of detection, sometimes being able to detect as low as nanomolar quantities, nanomolar concentrations of compounds, whereas for typical NMR applications, micromolar or millimolar concentrations of compounds are required, hence necessitating additional amounts of sample. But NMR has some additional advantages. One of those is that NMR is a non-destructive method, meaning that once you load a sample into an NMR tube, evaluate it by NMR, you can recover that sample at the end. The sample has not been destroyed, and hence one could recover the sample and conduct additional analyses on it. On the other hand, with mass spectrometry, after the sample has been subjected to the energy necessary for ionization, that molecule is destroyed. It cannot be recovered. And so that could be perceived as a con. Additionally, when we compare NMR and mass spectrometry, if we were desiring to avoid this mixture separation stage of having to use HPLC or GC to separate compounds within the mixture, NMR in some cases can be used with mixtures of compounds because of the fact that when we look at NMR data and we evaluate the x-axis, the ppm axis of a proton NMR spectrum, the different signals representing different chemical environments and different compounds, quite possibly, are spread across that x-axis providing us some ability to differentiate between compounds that are present in treatment versus control samples. On the other hand, with mass spectrometry, typically mass spectrometry is applicable only with pure compounds or relatively simple mixtures because the mass spectral data simply becomes too complex with more complex samples. And also, when a sample is directly injected into the mass spectrometer without prior separation by LC, there is not really sufficient selectivity to detect and evaluate the breadth of metabolites that would be present in a sample. And so mass spec generally requires that the sample be subjected to separation beforehand, whereas NMR, in some cases, this mixture separation step can be skipped. So what happens after the analytical data are evaluated? Well, then at that point, the process of analyzing those data begins. And so we'll put here at the end of our pathway the data analysis and interpretation. And that data analysis and interpretation can involve either untargeted metabolomics, looking for changes between treatment and controls in qualitative or quantitative terms. It can also look for targeted metabolites, meaning seeking a specific set of known compounds. For example, looking for metabolites that are present within the human metabolome database. So this would be the process of data analysis and interpretation. This is typically a very heavily computerized process because, as you can imagine, when comparing complete metabolomes between treatment and control samples, the data get very, very complex very fast and very difficult to simply analyze visually by visual comparison. And so there are a variety of software tools available to enable the comparison of data between treatment and control samples and pinpoint changes in the metabolome in response to particular treatments. And we will go into a bit more detail about the data analysis and interpretation of metabolomics data in the next video, so stay tuned for that.